because we really want to get this down, okay? All right, so first let's just start out with a question, which is, what's the smallest stable unit of matter?
Atoms.
Atoms, right. So atoms, everything, if we go smaller than an atom, we're getting into subatomic particles, and that is like nuclear chemistry. That's, you know, the domain of like atom bombs and such. So don't get into that with biomolecules, really. All right, and you're probably wondering, why the heck do we need to go into all this chemistry? I thought I was done with it, but of course, all these chemicals make up all the substances in our body, and we're going to be dealing with the physiology, right? We're going to be dealing with a lot of the interactions between the chemicals our bodies produce, such as hormones and enzymes. And of course, you're going to have a little bit more familiarity once again with all that lovely energy that we create, which is through glycolysis, the tricyclic acid cycle, also known as the Krebs cycle, and the electron transport chain. We'll get into that a little bit, I think on Thursday. So the chemistry is going to be dealing with all the physiology of how all these substances are interacting. And of course, when we get into digestion, all the food that we absorb is going to be chemicals. You know, we break them down. You'll find out that we break them down at the brush border, our foodstuffs, into smaller particles so that we can absorb and uptake at the cells, things like amino acids and sugars and small lipids, okay? And definitely all of our body substances are solvated in water. We're primarily water in our body. So we'll have to also talk a lot about the unique properties of water and what makes it such a great solvent. All right, so chemistry is the study of interactions between atoms and molecules. We'll see how these outer valences of electrons are pretty much where all of the big reactions are going to be taking place. The atom's the smallest unit. Of course, we will still talk about some of the subatomic particles, which is how we define every chemical element. Matter, anything that takes up space and has mass, okay? Having mass is different than having weight, because weight also accounts for the pull of gravity on the matter, all right? And when we talk about what's biochemistry from what's chemistry, is particularly this whole domain of reactions taking place in our body, the physiological processes. All right, so the atom. The atom is composed of negatively charged particles, positively charged particles, and some neutral particles that have no charge. So the negatively charged particles are the electrons that will circle in a cloud around the nucleus or the core of the atom. And in that core is where we're going to have the positively charged protons and the neutral neutrons. Oops. All right, so here in the core, right there, kind of nestled close together, positive charges, protons, neutrally charged neutrons, and then we'll build out all these valences or electron shells in which the negatives kind of are circling and moving all about the core. This positive charge of the protons kind of helps hold those electrons in close to the core, all right? So these are always kind of whirling about the electrons. Remember that like charges repel each other and opposite charges attract, right? So the opposite charges here kind of have a magnetic hold on each other. And here's sort of a diagram of the type of electron that we'll be dealing with. We can look here, electron, I should say atom. So again, positive charges, neutral charges, okay? Here's a core, right, the nucleus. When we have our first valence or electron shell that encircles, it fills up with 2 electrons. Can't take any more than that. Then we have the next outer shell or valence of electrons, and that can fill with up to 8 electrons, all right? We'll have a third one that comes out here, and that also can fill with up to 8. And then, you know, if we get into like going through the fancy metals and stuff, there's going to be all this other electron filling, but we're always going to deal with these first 3 shells pretty much in biological chemistry. Here is a chart from your book, just to remind you of what an atom is: electron, proton, neutron, their charges, what a molecule is, and what an ion is, all right? So we'll go through all that. All right, our chemical elements are going to be atoms that all have the same number of protons. This is what defines the atom. All right. Can you hang on one second? I'm getting, my mother is calling me and my health attendant isn't here. I'm really sorry to interrupt like this. I'll just stop. Hello? I'm teaching. Quick. Okay. Can you come and take care of mom? She's calling and talking still not here, and I Bye. What do you need, Mom? Chris is coming, okay? I gotta teach. I'm in the middle of teaching. Chris is coming. Okay, sorry about that, everyone. It's kind of an emergency. My mom is 95 and she has a UTI right now, and she's a little bit delirious. And my nurse's aide was supposed to show up and didn't. So my sister is rushing over here to take care of her. So I apologize. All right, so atoms with the same number of protons are what determines an element. So the number of protons in the nucleus, if you have 1, it's hydrogen. It's always hydrogen. If you have 2 protons in the nucleus, it will be helium. It's always helium. It's nothing else. If you have 3 protons in the nucleus, it's lithium, only lithium, and nothing else. And that's kind of the way that it goes, all right? Determines what that element is, all right? We'll see later on the variations in the electrons determines the chemical reactivity. We can add more or less neutrons into the nucleus, but it doesn't change anything but the weight of the whole atom or the weight of the whole molecule if we're dealing with molecules, okay? It's not going to change what substance it is. So the number of protons becomes what we call the atomic number. So hydrogen has an atomic number of 1 because it has 1 proton. Helium has an atomic number of 2 because it has 2 protons. Lithium has an atomic number of 3 because it has 3 protons, and so forth. All right. If you change the number of protons, you change the number of the atomic number, you're changing what the element is. Now, here you can see that we have atomic mass. Oops. Atomic mass of 12, 13, and 14. The symbol is C for carbon. So these are all carbon. So they all have the atomic number of 6. This means they all have 6 protons. So what's the difference? What's the difference of changing this atomic mass? It's the number of neutrons, okay? So if the atomic mass is 12 and you have 6 protons, you then have 6 neutrons. We give each neutron and each proton 1 mass unit. The electrons, which are so much smaller, negligibly add to the weight that we basically say their weight is 0. Okay? So when we have a carbon, atomic number of 6, but its atomic mass is 13, what has changed? Anybody want to tell me?
The neutrons.
And how many do we have?
7.
7, right. Okay. Carbon 14. We have how many neutrons?
8.
8.
Atomic number is?
6.
And the number of protons are?
6.
6.
Okay, great. And we'll kind of being repetitive here because of course, when you get into testing situations and, you know, these are the things that are easy to forget, but it's a relatively easy concept, right? All right. And so here you can see in the isotopes of carbon, you can see that we have the same number of electrons out here in the shells, okay? You'd have the same number of protons that are the green. It's going to be these bulky white ones that are kind of hard to see where you had the variation in the neutrons. Chemical reactivity is going to be the same for all these. Carbon will undergo the same reactions, whether it's carbon 12, carbon 13, or carbon 14. And we can use this when we want to like track carbons. We can use an isotope that's, you know, relatively rare and put it like in the body as a tracer and kind of track where it goes, okay? We'll talk about that in a little bit. All right, so the atomic mass is the number of protons plus the number of neutrons, reminding you that the electrons don't contribute to the mass because they're so light. Isotopes vary by the number of neutrons. And for any element, when you read on the periodic chart, and you'll see that carbon, the atomic mass is not just 12, it's like 12.01. It's because we take the average of all the naturally occurring carbons. So we have carbon 12, carbon 12 will be like 99.7% of all the carbon. And then there will be small contributions from carbon 13 and carbon 14 that add that 0.01 in there, okay? That's how we get the funky number that you see that's not exactly a whole number. All right, so isotopes of oxygen, oxygen 15. Oxygen always has 8 protons, so oxygen 15 has 7 neutrons. Oxygen 16 has 8 protons because 8 protons defines oxygen, so it has 8 neutrons. What would be the composition of oxygen 17? Anybody?
8 protons, 9 neutrons.
Yeah, I know. Seems ridiculous, but to keep going over it in depth like this, but we need to just to point it all out. I don't think you guys.
I have a quick question. If it's too like off topic, let me know. But why are electrons so light?
Why are they so light?
Yeah, they just are.
They just
Okay.
are, yeah.
They just are.
They're
Okay.
very small particles. They're basically like a flying around charge. You know,
Okay.
they're just really, really light
Okay.
reactivity.
Okay, yeah, I had missed that like while reading for some
Mhm.
reason, so I didn't visualize it correctly. So I was just,
Okay.
thank you. That
Yep.
helps.
All right. And you know, and you can see here, this is what everything looks like on the periodic chart. And like I said, you know, normal hydrogen has 1 proton in it, but there's the heavier hydrogens like deuterium and tritium. We give the hydrogen ones special names, and they kind of average in that 99.985% of all hydrogen is hydrogen 1. Okay. And so you can kind of look at that chart if you want to kind of, you know, work with all that in your mind if you're having trouble. All right, so what's the whole point of all these heavy isotopes? Well, heavy isotopes have an extra neutron, right? And they're a heavy isotope. We call it just a heavy isotope if it has the extra neutrons and it stays stable. It has a nice stable nucleus. Nothing's, you know, going to change. It's not going to emit radiation or anything. So something like carbon-13 is a heavy isotope. It has an extra neutron in it. And it's just a little bit more mass. It's not radioactive. Radioisotopes, however, have an extra neutron and the nucleus is unstable. It doesn't like having that extra neutron in it. And so it's going to want to decay or emit some particle out of the nucleus. And we don't want to get too in depth into, you know, getting into the physics of this. But just for our intents and purposes, and you don't know, you'd have to be told which ones are heavy isotopes and which ones are radioisotopes. You know, you're not going to be able to predict that. So carbon-14, which is a really minor amount of all carbon and what we often use with carbon dating, is a mass of 14. So it has a second extra neutron in the nucleus. And this is going to lead to an unstable nucleus that decays. Okay. So what is radioactive decay then? It's going to be the loss of 1 or more neutrons to release that energy, kind of get rid of that tension going on in the nucleus. And it's released in the form of things like gamma rays, alpha particles, and beta particles, which depends on the isotope. And that, like I said, gets beyond, we're not going to get into physical chemistry for that. So just know some isotopes are heavy, some are radioactive, and we'll use both of them in medicine for different purposes. All right. For research, for tracers, and, you know, to do things like treat with radioactivity. All right. So all chemical elements form isotopes, right? Atoms will form bonds and interact with other atoms based on their electrons. So the various isotopes of an element still have the same electrons numbers encircling it. So all their reactions will be the same. Carbon-12, carbon-13, carbon-14 will undergo the exact same reactions. The number of protons, the number of electrons are always the same. It's only the neutrons that vary. Isotopes of an atom will thus chemically react in the exact same matter. And they're going to serve the same functions in our body for all the metabolic reactions, all the series of chemical reactions. They can be stable or unstable, okay? And remember, stable are just the heavy isotopes we can use as tracers. The unstable ones are radioactive. Radioactive isotopes are unstable, but typically when we're talking about isotopes and the form, you know, when we're looking at all of our chemical equations, we're usually talking about what's the most stable element. So when we talk about carbon and we see, you know, the atomic mass is 12.01. We know that carbon 12 with 6 protons and 6 neutrons is the most common one in nature. It's the most common, it's very stable, it's not radioactive. And so that's what we're usually going to deal with, except when we talk about the ones that are radionuclides that might be used in something like a PET scanner, okay? And we'll talk about a little bit about how when the molecules or the atoms decay, they have half-lives while they're emitting that particle that they're trying to get rid of out of the nucleus until they reach a stable state. So you can have, you know, a half-life, if you had a half-life of a week, all right? That means that at the end of the week, 1/2 of whatever amount of matter you were dealing with, however, whatever number of atoms would be decayed. Okay, so if you start out with 200 atoms, at the end of the week, you would have 100 atoms, okay, that have decayed. All right. All right, so here's an example that was in your textbook of how we use radioactive substances. Radioactive iodine, 131. So my sister had to do this. She had to go and take radioactive iodine. She had a hyperactive thyroid gland, a little thyroid nodule, and they needed to destroy the thyroid tissue. Thyroid tissue is the only tissue in the human body that takes up iodine. So in order to destroy it, what we can do is put in radioactive iodine, and it will get taken up in the tissue only of the thyroid gland. It's radioactive, and the radioactivity can then destroy that portion of tissue. Okay, so you don't have to undergo surgery. All right. So she could take a little radioactive iodine pill, and she had to go like sit in a room that was, you know, kind of lined with some paper and stuff. And you had to be there for like 3 days until the half-life had decayed enough that it was going to be safe for you to go around other people. And then somebody comes in and cleans up, you know, in a spacesuit, comes and cleans up the room for any leftover matter or any, you know, clean everything up. So we use radioactive isotopes in that way. Sometimes, like in that case, to treat tissue we want to destroy, like maybe destroy a cancer. We can put little radioactive iodine pellets. We can use them to like track things like when we want to track the coronary blood vessels to see if they're clogged, we can put some radioactive tracers and see if the blood going through flows, you know, in a nice laminar pattern, or if it's kind of tortuous in a narrowed coronary vessel. And we can have a little detector that detects that and makes a picture on a screen for us. So this is great stuff for bio tracers. All right. So our elements, which are defined again by their atomic number, how many protons they have, are the simplest matter with having a specific chemical property. And we have 92 naturally occurring elements. Compounds are formed when we combine different elements. Okay. A molecule is also a combination of different atoms. We can call it just a molecular particle, a molecule, if it has 2 of the same atoms, 2 or more of the same atoms, like H2, 2 hydrogens. It's a compound if it has 2 different atoms, H2O, water, compound, okay? A compound is also a molecule. Water is a molecule, water is a compound. Molecular hydrogen, H2, is a molecule, but it is not a compound because it has the same atom, okay? All right, bulk elements. Sometimes we require bulk elements in our body. These are the elements that we require large amounts of. Carbon, the backbone of all of our organic materials. Okay, so lots of carbon in your body. Oxygen, obviously, it's what we use to respire. Hydrogen, okay, hydrogen and oxygen together, water, kind of the main solvent of our body. Nitrogen, which is going to be one of the elements that makes fundamental for all of our proteins and also fundamental in all of our DNA. And then sulfur and phosphorus, we also have in large number. You know, phosphorus in, for instance, our ATP. We'll also have a lot of trace elements, and these are usually important as like catalysts or key to various biochemical reactions that take place. All right, so trace elements are going to be things like iron and iodine. Ultra trace elements are elements that we use in really minute amounts. Things like arsenic or gold. We actually have little trace amounts of that in our body, and it's normal. All right, and here is a chart that goes through, I think this is from the old version of the textbook, but I kept it in here just if you want to look at it. These are the bulk elements in our body, and then you can see some of the more trace elements as you go along. All right, so all of our atoms and molecules, our atoms have the electron shells. We talked a lot about the nucleus. Let's get into those very lightweight electrons. The first shell or cloud or valence, you can use all those words interchangeably, of the electrons that float around the nucleus is going to hold up to 2 electrons. Hydrogen, which has 1 proton in it, will have 1 electron. And to be a neutral atom, you have to have the negative electron charge balance the number of positive proton charges in the nucleus, and they have that attraction to each other. So 1 proton plus charge in the nucleus, 1 electron negative charge. The next element, helium, has 2 protons in the nucleus, so 2 positive charges. It will therefore have 2 negatively charged electrons to balance it. Okay, that fills up that inner shell that only takes 2 electrons. As we add additional protons, as we go across the periodic chart, next going to lithium, which is 3 protons, we now have to move into the next bigger shell. And remember, that takes 8 electrons. So lithium will have 3 protons in its nuclear core. It'll have 2 in the innermost shell called the S shell. And the next valence or shell or electron cloud out there is going to have 1 electron, okay? And we can go across, I have a picture here of the periodic chart. We can go across and we'll keep filling in more and more electrons as we fill in more and more protons, all the way up until we get to 8 electrons, which then takes us to the next, what we call noble gas in the periodic chart. That means the outer valence is completely filled. So you'll have an inner valence with 2 electrons and an outer valence with 8. The outer valence is where all the action takes place for forming chemical bonds. So if there's space in the outer valence, it's going to be looking to fill itself with further electrons. It's going to be looking for partners to kind of bring some electrons into that valence. If the valence is already filled, it has no incentive to partner up. And that's what we call the noble gases. And that's at that last column of the periodic chart. Let me see if I got a period. Here it is. Okay. So going across, you see hydrogen has 1 proton on it. It will have 1 electron. Helium has 2 protons in the nucleus, so it has 2 electrons in that S shell. Lithium has 3 protons as we go across, 3, 4, 5, 6, 7, 8. These are the atomic numbers. So that tells you the number of protons. It's what defines the element. Beryllium has 4 protons in it. So it has 2 in the inner shell and 2 in the outer shell. Now here, when we've gotten down to this second row, you have to assume that it's helium with an inner shell filled with 2, and now it's just showing the outer valence. Okay, so we've moved up to that next outer valence. Boron has 3 because we have 5 protons in here. And remember, there's always a 2 inner valence of 2 electrons here. Carbon with 4 in its outer valence, nitrogen with 5, oxygen with 6, fluorine with 7, neon with 8. This going down the column, these all have 8 in the outer valence. These are the noble gases. When we go down a column here, these all have 7 electrons in their outer valence. We call these the halogens. Fluorine, chlorine, you'll see iodines in there, all right? Oxygen and sulfur, these have 6 in their outer valence. Nitrogen, phosphorus have 5. Carbon has 4. Silicon has 4. All right. And so forth. Lithium 1, sodium 1. Okay. All right. So each shell has its maximum number of electrons that it can hold. The inner shell always holds 2. The next shell holds 8. The third shell holds 8. The electrons will be in electron pairs. All right. Now, when we draw these things, it looks static, like it's all just sitting in a place. But you got to remember, the electrons are always whirling all about. Okay. They're moving, constantly in motion. And when we fill these, we fill them unpaired first. Okay, so let me get here and see if I can draw it. Just draw in here. All right, so if I were saying I'm going to fill, doesn't want to let me draw. Okay, so if I wanted to fill, let's do oxygen. Okay, oxygen has 2 in its inner shell, right? 1 and 2. And then in its outer shell, what does it have? Anybody? It has 6.
6.
Yeah. So you would fill it unpaired first. 1, 2, 3, 4, 5. I'll do here 6. So then you see there's one unpaired electron and another unpaired electron. And this is why sometimes you'll see oxygen written with, whoops, like unpaired electrons, or sometimes more commonly, you'll see oxygen written showing its paired electrons like that, okay? And you'll see that it's making bonds where it wants to bond this electron and this electron with another element, okay? Or another atom.
I have a quick question just in terms of the order in which you drew the electrons. While reading, it seemed like the 1, 2, 3, 4 were critical in where you placed them, but then the 5 and the 6, I feel like I've seen them placed in different, like, do you always start from the bottom? Or
you mean like, could I have done instead, let's draw this circle around here. Could it, instead of putting it down here, could I put it over here?
Correct. Yeah,
Yeah,
that's what I mean.
Exactly.
Okay. Okay.
Yeah.
So once
Yeah.
you're done with the 4, the 5, 6, 7, or 8 are kind of optional in terms of where you put the circle. And oftentimes, like, the illustrator, whoever's drawing it, will put it closer to whatever other bond it's trying to make. I don't know
Right.
about that, but.
Right.
but-
Well, and that starts getting into the decisions on that a little bit into a level of chemistry we really don't need to get into. But what happens is that unpaired electrons, and when you form the bonds, the molecules, like if we took carbon, you'll see it kind of always drawn kind of like this.
Yeah.
And maybe like, because it wants to form a tetrahedron. So
Okay.
but each of the pairs of the electrons wants to space themselves out. You know, it's all a big circle, right?
Right.
So they want to space themselves out as far as possible from each other. And so the bond angle in between tends to be about 109 degrees.
Okay.
Okay. We're not really going to get into bond angles and things like that. But yeah.
Okay. Thank you.
You're remembering something that's
Okay.
it's really important when you get into organic chemistry, but for our intents and purposes, we don't have to focus on that. So that's why I'm leaving it out.
Okay. Thanks.
Yep. Because we don't want to start making it harder and more confusing than it has to be. All right. All right. So getting back to this. So what we find then is everything gets organized in, as well as going across the rows, we're adding in protons and adding in an electron as we go across the rows. We call it the periodic chart because there's trends. Group 8, all these are the noble gases. Their outer valence is filled with 8 electrons. So they're unreactive, okay? And they all behave pretty much the same. This row, group 7, is deficient in 1 electron. If you can add in an electron, it'll look like a noble gas. And that's what all these want to do, is they want to ultimately try to look like a noble gas, okay? So since it's deficient in 1 electron, it's going to search for some way to either steal an electron from another atom or share an electron with another atom, all right? And you'll find all these halogens kind of behave the same in the way they want to steal or share electrons. Same things with, oxygen, sulfur, selenium. Same thing with nitrogen and phosphorus, carbon and silicon, okay? So we're going to be concerned largely with, whoops, let me draw again. We're going to be concerned largely with the elements that are sort of up here and kind of around here. Really not xenon or kryptonite, okay? Sort of that. All this stuff down here, we'll rarely ever talk about any other of these atoms in our biological. Occasionally lead if we want to talk about lead poisoning, or barium when we talk about X-rays, and we do things like barium enemas and that, but we're really not getting into that, okay? All right. All right, so now, because our molecules are deficient in electrons and they want to find other atoms that they can steal electrons from or share them with, they're going to try to steal or share and combine in different ways, okay? So to form molecules, 2 or more atoms combined. Like I said, a compound is when 2 or more atoms of different elements combine. And we'll have to learn to write molecular formulas. So a molecular formula would be H2, 2 hydrogens bonded to each other. We'll also see that a molecule of glucose, in which we'll have a chain of 6 carbons surrounded by hydroxyls, OHs, will be written C6H12O6. We'll get to more complicated stuff like that down the road. Or water, 1 hydrogen and 1 atom of oxygen. That is a compound. It's also a molecule, okay? We'll talk a lot about water. All right, so here you see two hydrogens combined with each other. They're sharing their electrons. All right, so when they're sharing their electrons, you have your hydrogen with its one electron, and you have another hydrogen with its one electron. Neither one is really motivated to give up its electron to the other atom. So what they do is they share it, and that's a covalent bond when they share it. And we write it as H with one single bond between the two. And your chemical notation is, of course, H2. Same thing with O2. If we had our oxygens, we had the inner shells filled, right? And then we had our outer shells where we had 1, 2, 3, 4, and then we had, I'm going to put them out here, 5 and 6 double pairs. It's kind of messy, I'm sorry. Again, we'll do another oxygen over here, inner shell. Well, I'll make that the outer shell because I made it so big. 1, 2 inner, 1 outer, 3, 4, 5. Let's put that one here and this one here. And see, part of why I drew it this way is because then I can say, I'm going to share this one with this one, and I'm going to share this one with this one. Okay. And then I have my two lone pairs on the outside. So I have oxygen sharing with oxygen, and then I can put my lone electron pairs on it if I want to remind myself that it has lone electron pairs. And that's what this would look like. Really, they would have their lone electron pairs drawn in out here. And you'll see why these lone electron pairs become really important, okay? All right, now, I said sometimes we share, but sometimes we like to steal and just strip another atom of its electron. So an atom that has either gained or lost completely an electron is going to be called an ion, okay? So if you gain an electron, you now become negatively charged and you're called an anion. If you are stripped of an electron, you lose a negative charge and become positively charged, and you're called a cation. So here I'm talking about a neutral sodium atom. It has 11 protons and 11 electrons, all right? Whoops. So if we were to draw in sodium, okay, and we had, let's just write 11 protons and put a little plus symbol right in there, and that's its core. So it would have 2 electrons out here, and then in its outer shell, we would have to draw 9, right? 1, 2, 3, 4, 5, 6, 7, 8. Going on to a third shell, and 9. So it has 1 lone electron out there in a valence. Now, when you look at sodium, its strategy is going to be, is it easier for me to give up or donate 1 electron or somehow find 7 other electrons out here to fill the shell?
Donate 1 electron.
Donate 1 electron. So you're going to find that sodium and all the atoms in the same column that have 1 electron as a lone electron in its outer valence tend to give up their electron and become positively charged cations. So it becomes Na plus. I wrote the plus in here when I shouldn't have already. Sorry about that. Just have it. You'll almost always see sodium as the cation, okay? And cation, you can always remember which one's cation and anion, as the mnemonic device of cations have the positive charge in the word, okay? All right. All right, so our chemical bonds are going to form when these atoms combine. I already showed you sort of one covalent bond, and now I showed you what an ion is in the terms of the sodium cation. So we'll see ionic bonds, covalent bonds, and how they form with each other, all right? All right. All right, so in an ionic bond, when sodium forms a cation, it now has a positive charge, okay? It's transferred or given away its electron to some other atom. And that atom that it gives it to takes a new electron, and it thus becomes negatively charged. So now sodium plus the new negatively charged anion will have an electrostatic attraction because opposite charges attract. So they'll kind of like magnetize to each other and want to stay closely associated. The electron really being with the one electron being with the new, say, chloride, and being gone from sodium. Okay, this makes the two kind of unequal in electron content, but they have that attraction. So we say this bond is ionic. It has a lot of ionic character. All right. Sodium is the cation, the electron donor. And in the case I'm talking about, chloride will be the electron acceptor. All right. Now, when we go back, if I look at this, this periodic, boy, I moved that mouse, it wants to change. If I look at this periodic chart where you have lithium and sodium, sodium always wants to give up this electron, and chloride over here has 7 electrons, so it's always looking to get one. So what you're going to find is that you're going to have natural ionic bonds form between the elements or atoms of column 1 and column 7 here, okay? So they're all going to do this donation thing of sodium's electron going over here, okay? So you'll see sodium, now a cation, and chloride, negative, right? Makes sodium chloride. You could also have sodium fluoride, NaF, where fluoride would be the anion and sodium is the cation. You could have lithium chloride and so forth. All right, that's the easiest one to examine. Oh, shoot, sorry. Hit the wrong button. All right, now, when you're seeing the electrons orbiting the nucleus, just remind yourself, let's review here, stop for a minute. One proton identifies the element, makes it hydrogen. The one proton in the nucleus has to be counterbalanced by one electron flowing around it to be an electrically neutral atom in its, home state form. Helium has 2 positively charged protons, and it has 2 balancing negatively charged electrons. This is a filled electron valence or electron shell. Helium also has additional weight in atomic mass units from neutrons that add weight but do not add charge. When we move up to the third element, adding in a third proton, we're at lithium. Its new electron, it has 3 electrons balancing the 3 protons. The 2 in the inner shell held most tightly and most closely. Now this outer valence is a little bit less tightly held. You know, the cloud is a little bit farther out, and that's partly why this is the electron that's easily stripped off of lithium or easily donated. So lithium's going to act like sodium and tend to donate that electron. Okay. Here's the sodium atom, has 11 protons, 12 neutrons, and again, it has 11 electrons, just like lithium, 1 in its outer shell. And that 2 farther out will easily get stripped away to form the cation. In the cation, the ionic bonds will form because of the opposite charge between the one molecule that completely donates its electron attracted to the one that has gained it and now has a negative charge. Okay, so the formation of the sodium ion, you have the electron being stripped and it's going to be accepted over here by chlorine. So then we see sodium now has a positive charge and is down to just the 2 electron shells. And we have chloride now with its filled 3rd electron shell out here. Okay, positive charge, negative charge, ionic bond. They like to hang out right next to each other. Okay, here it is again, sodium chloride forming. And what you find is that in salt, sodium chloride, they like to line up and they form crystals in a set arrangement. Okay, so that this sodium in here is always kind of associated on one part by a chloride, associated with a chloride over here, associated with a chloride at this end of it, associated with a chloride kind of surrounding. Okay, and if you look at it in terms of chloride, each chloride is surrounded by a sodium molecule, okay, or sodium atom. Okay, sodium here again, losing its electron, positively charged. Chloride will gain that electron right here, chloride ion. I kind of have it written out. Some people look at these things better going across, you know, some people like the pictures. All right, so the ionic bonds, complete attraction, associated together, opposite charge attracts. Sometimes in biological textbooks, they make you believe that ionic bonds aren't strong, and I'll explain why that is. They are exceptionally strong bonds. If you have salt sitting on your table, it will sit there and be salt forever. It takes a lot of energy to get in there and destroy the bonds of sodium chloride. It has a really high melting point. So don't think that these are weak bonds. There's always kind of a little misnomer when a lot of biological textbooks say what's the strongest bond and they want you to pick covalent. What happens is that these salts, the ions, can dissociate when they're in a solution. And you know this, if you take sodium chloride and you put it in water, it's no longer the salt crystal. It dissolves, it solvates. And we'll go through why. All right. All right. So sodium chloride, strong ionic bond. All right. They kind of associate living next to each other because of their opposite charges. Same thing happens here. The outer valence has the highest energy and it's where the bonds are going to form. Lithium is just like sodium. It has the 1 electron out in its outer valence, and that's the electron that it will tend to lose. We also write these in what we call Lewis dot structures. In a Lewis dot structure, we write the element with the outer valence electrons kind of surrounding here. You could see that a filled electron valence would have 2 dots here, 2 dots at the bottom, 2 dots here, and 2 dots here. Like if I were writing helium in a Lewis dot structure, or like fluorine, as we have right here. You can see fluorine has 7 electrons surrounding it. I write it with the 7 electrons, and I see where my unpaired electrons are. So you'll see those Lewis dot structures are very convenient. And you can see that just like sodium chloride, lithium with its 1 lone electron, and fluoride, which needs an electron, will do exactly the same thing like sodium chloride and form an ionic bond, transfer completely an electron, and you'll have Li plus F minus. Okay, so that's ionic bonds. Covalent bonds, we share an electron. Like I said, the textbook says that they're stronger, but what they really mean is that when you put things with covalent bonds into solution in water, the covalent bond doesn't just drift apart like the ionic bonds. The sodium positive and the chloride negative will move apart, okay? And we'll see this as we get into the properties of water, all right? So chemical bonds, covalent bonds share electrons, all right? And we'll see some bonds, they'll share the electrons pretty equally. And in other bonds, yeah, one of the atoms will kind of be a little bit more greedy and keep the electron hovering about its nucleus a little bit more than the other one, okay? It's a little bit of a dysfunctional relationship, right? All right, so we have a giver and a taker a little bit. Our ionic bond, we have a completely dysfunctional. Somebody is really, the giver and somebody is the taker. Covalent, that's what we all want in a relationship, equal sharing. And then we'll have some covalents that are a little ionic-like, and we'll call them polar covalent bonds, all right? So let's look at how that happens. So with our hydrogen atom, we showed this before, each having 1 electron. They're the same atom, okay? So they're going to share entirely equally. There is no reason why this electron and this electron are going to want to dance around the nucleus of this hydrogen atom any more than this one. So they're all going to kind of whirl about encircling the nuclei of each of these atoms equally, all right? Now, when we get into a water molecule, we see here some hydrogen atoms. Here you see an oxygen molecule, O2. O2 is the same. We have oxygen, oxygen, double bonded to each other. And all these electrons, no reason to dance about this oxygen more than this oxygen, right? They're the same. When we get to water, you can see the difference. Oxygen is a big atom, right? A big atom with 8 positive charges in its nucleus. Hydrogen is a small atom with only 1 positive charge in its nucleus. So these electrons out here that are being shared between hydrogen and oxygen are going to be a little more attracted to all that positive charge in the big giant nucleus of oxygen. So they'll spend more of their time dancing around oxygen and a little bit of time dancing around hydrogen, okay? We call this more electronegative, attracting the negative charges. Oxygen is a more electronegative molecule, atom, than is hydrogen. So this is what makes a polar molecule. More of all these negative charges will hover about the oxygen in a water molecule. So we always say then that oxygen will kind of have, in a water molecule, will have a slight negative charge to it, okay? And when we say it has a slight negative charge to it, we'll write it as delta negative, all right? And the hydrogens will have a little slight positive charge to it. So we write that as delta positive, delta meaning partial, okay, partial charge. Is this making sense to everybody? Okay.
A little bit.
All right. So a covalent bond, when hydrogen and hydrogen each only have one proton, and when they share an electron between them, okay, and they have no reason why, you know, why would this one be able to strip an electron away from this hydrogen? They're the same thing. They have an equal attraction or equal ability to hang on to their own electron. So you see, so they share the electrons. But like I said, one proton here, one proton here, nothing is drawing this electron more toward one part of this new molecule than the other. So they share it equally, and this is called a nonpolar, okay, covalent bond, equal sharing. You know, this is like the true relationship we all seek, where everybody shares and shares alike, and everybody's kumbaya, all happy, 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 right? All right. Carbon and 4 hydrogens, okay? Carbon has has the 6 protons in it. It's got its 2 s electrons, and then it has 4 electrons on the outside. All right, and it bonds here with 4 hydrogens. Well, why are these electrons going to spend any more time around this hydrogen or this hydrogen or this hydrogen or this hydrogen? It's not. All these hydrogens are pulling those electrons away in equal, like, vectors or directions. This too is a non-polar, no charge anywhere on it molecule. Okay, so non-polar covalent. When we get to water, and we'll look at some other molecules, let's just kind of go back here. If I want to make up a molecule and say, let's look at carbon dioxide. Okay, so carbon dioxide, if I were to draw it on, let's see how we're going to figure this out. This is where I really wish I had a chalkboard. Let's just add in a slide first. Oops, what'd I do? Let's add a blank slide here somewhere. All right, so carbon dioxide. So we're going to have our carbon, okay, which we know has 2, right? And then it has 4. Sorry, I just can't control my drawing with the mouse that well. All right. Now, if I have oxygen, oxygen has 2, right? Inner. And then its outer has 1, 2, 3, 4, 5, 6. Same thing down here. Oxygen. One, 2, 3, 4, 5, 6. How is carbon going to combine with these? Okay, so it's going to form a sharing bond between this one here, right? And let's say, well, I probably should have drawn this. Erase one. Whoop, erase the wrong thing. Sorry about that. And this one and this one, a double bond, okay? And it's going to double bond between this one here and this one here. Remember, this has 6 positive charges in its core. Oxygen has 8 positive charges in its core. Now, what's going to happen with its shared bonds? Are these shared electrons, are they going to kind of want to hover more about oxygen or hover more about carbon, do you think?
Oxygen?
Oxygen?
Oxygen, yeah. Because oxygen has 8 positive charges in its nucleus, right? So it's going to pull these electrons toward it more strongly. And sometimes you'll see the drawing for it. They'll kind of draw the vectors on it that way. All right. Pull the electrons more strongly toward that oxygen. Pull the electrons more strongly towards this oxygen. So if we were to put it in there and look at our carbon dioxide, it's going to be C double bonded to oxygen. C double bonded to oxygen. You could draw its lone pairs on it if you wanted to. Okay. Lone pairs on it. And it's going to be polar because we're going to have a little bit of a negative charge out here, a little bit of a negative charge out here. This is going to have a little bit of a positive charge as we, let's put a different color. As we kind of draw the electrons away, right? We're kind of stripping electrons off of that carbon. So that carbon is kind of like a little bit positivity to it. We call that polar. So it's covalent, it's shared, but it's polar. All right. All right. All right. So when we want to write our molecular formulas, covalent bonds, we write with the lines in between. Okay. You don't draw the lines in between for ionic. You just write NaCl. Okay. That they're closely associated. Like I said, you can always draw in the lone pairs of electrons. You can draw in the delta pluses, delta minuses on the polar. Is this polar or non-polar?
Non-polar.
How about this?
Non-polar.
Yep. This?
Polar.
Yep. Polar. Okay, good. All right. So summarize that. Non-polar covalent equal sharing, right? Electrons have the same negativity. Protons are not going to pull, there's not going to be some molecule that has like more proton heavy pulling the electron away. Polar covalent bonds, unequal sharing, okay? Because one of the atoms has a disproportionate pull on the electrons. It might have, a boatload of protons there in the nucleus pulling the electrons toward it. And this would be molecules like water, carbon dioxide, all right? And we think of it as molecules that have a little bit of ionic character to the covalent bond. And what you find is that electronegativity, we'll talk about it, is describing the tendency of the atom to attract the electrons toward itself. They have a more positive core. They tend to have more protons. And these tend to be more electronegative atoms over here. These tend to be more electropositive. So these are the atoms that tend to draw the electrons away when they are in covalent bond relationships. All right. Okay. So non-polar covalent bonds, again, found between atoms with about the same electronegativity, if you want to use that chart. Polar covalent bonds are when you have them at opposite points on the chart. Oxygen, and the ultimate pull electronegativity is when you have something like fluorine and lithium. It just completely pulls it away, that it actually strips it, steals it, and it's in an ionic bond. All right. So sometimes we're taught to think that the ionic bond and the covalent bond aren't related or they're completely different, but it's all really on a spectrum. Okay, so we have the ionic bond where it really does strip it away. Okay, and one atom basically loses an electron and the other one gains it. Okay, so there's no real sharing. It's spending all of its time around the anion. At the other end of the spectrum, we have the nonpolar covalent bond where the electrons are dancing about both nuclei, both the cores of each atom, okay, equal sharing. The polar covalent bond, you think of as somewhere between this ionic bond and the nonpolar covalent bond, where it's covalently bonded, but really kind of a little bit ionic character, stripping the electrons away so that you have one molecule that's more electronegative and one that's electropositive. And this will become important when we think of acids and bases, because a lot of your acids, your proton, your hydrogen in the molecule will basically be just a proton, okay? Just a proton. All right, so when we talk about all this and we talk about water molecules in particular being our universal solvent, you know, it's the solvent of our body. All of our blood is made in water. We always have our slightly negative end of our oxygen and our slightly positive ends of our hydrogens, okay, written out here, delta negative and delta positive, because we're spending almost all of our electron cloud dancing around the oxygen atom. And because of this, water, when one water molecule is next to another water molecule, is next to a water molecule, is next to a water molecule, such that you have a pool of water or a beaker of water or a cup of water, the water molecules, one next to the other, will form what are called hydrogen bonds, okay? So here's what hydrogen bonds look like. These are, you have to think of them as an attraction. I hate that they're called hydrogen bonds. I feel like they should be called hydrogen attractions, okay? Because they aren't really like bonded in the sense of either our ionic bond or our covalent bond, but the molecules will always align up such that the negative, partially negative end of oxygen will try to face a partially positive end of the hydrogen. Here, a partially positive hydrogen again faces a negative oxygen. And you could draw going on and on till it has this lattice-like structure. And this hydrogen bond formation is what's going to give water all of its unique properties. Because while every attraction here of these hydrogen bonds in and of itself isn't very strong, the sum total of every water molecule in a beaker lining up and forming this lattice-like structure results in lots of attractive forces. And this is why water has a high boiling point, okay? It's also why water has the cohesive properties that it like kind of clings to and creeps up a glass and forms a meniscus. It's why when water falls on your car, it beads up. It doesn't just run all off, you know, and it pools up, okay? High boiling point, all these great things that give water its characteristics because of the way it aligns one molecule after another. All right, so let's take a little mental break here and see if we're getting all these concepts, all right? So the type of bond formed by sharing electrons is which one?
Covalent.
Yeah, okay, covalent. All right, now at room temperature, we have the gas of oxygen and neon. Oxygen combines with other elements, but neon does not. Why would that be?
Because neon has 8 electrons filling its valence shell.
Yeah, and oxygen has 2 spots empty, right? It only has 6. Okay, good. All right, next one. Which element only has 1 proton in its nucleus?
Hydrogen.
Yep, Okay. And the innermost electron shell holds how many electrons?
2,
2,
Okay, what about the next one?
8,
What about the one after that?
8,
8.
Okay, good. The smallest stable unit of matter is?
atoms.
Atom.
Okay. And 2 atoms that are the same combined form a,
Molecule.
2 atoms that are different combined form a
Molecule.
molecule, and also a
Compound.
Compound.
compound. Yep, okay, great. The atomic weight or atomic mass of an atom reflects the average number of,
Protons
Protons and neutrons.
what about the electrons?
They're too light.
All right, negligible. The nucleus of an atom consists of,
protons and neutrons.
same thing, right? Okay. Isotopes differ in the number of,
Neutrons.
yep, neutrons, okay. And when electrons are transferred from one atom over to another atom, the 2 atoms then unite due to opposite charges. We call this,
Ionic bond.
ionic.
oh, you guys are so smart. If an isotope of oxygen has 8 protons, 10 neutrons, and 8 electrons, its mass number is
18.
18, right? What's its atomic number?
8.
8,
Good, okay. All right, and now we got chemical reactions, okay. Chemical reactions are basically going to involve the making or breaking of bonds between atoms, and bonds store energy. So if we want to break apart bonds, we're usually going to have to bust them up with putting energy in there to bust it up, okay? So a change in chemical energy is what's causing the change in a chemical reaction. We call it an endergonic reaction when all the reactants are absorbing energy, okay? And they're making like a new molecule that probably holds a lot more energy in it. We call it exergonic when the bonds are breaking and it's releasing energy. Whoa, going back, okay. So synthesis reactions. Synthesis reactions are when we're going to form a new larger molecule from 2 smaller, simpler. So here we're going to add A plus B, and we're going to build it up into molecule AB. Sometimes we'll call these condensation reactions. These are part of anabolism. So when we talk about like cellular metabolism, there will be anabolic and catabolic reactions. Anabolic is always building something bigger. Okay, so anabolism will be synthesizing bigger molecules in our cells from smaller molecules, such as taking simple sugars and kind of knitting them together to make starches, carbohydrates, polysaccharides. All right. Decomposition reactions are when we want to split a molecule into its smaller component parts. It's kind of the opposite of the synthesis reaction. So we're taking AB and we're going to split it into A and B. This would be catabolism. You also do this in the cells of your body, like when you digest your food and you have enzymes coming in, and you're going to take that big carbohydrate donut you just had for breakfast and split it down into its simple sugars so they can get absorbed and taken up by the cell in the small intestine. This is catabolism. All right, so decomposition reactions, catabolism, breaking chemical bonds, and we usually in the body do this as hydrolysis reactions, meaning we use water to come in and split that bond, okay? So we have AB, we add some water to it, and we end up splitting it into A and B, where A has a hydrogen attached to it, and B has a hydroxyl. OH is called a hydroxyl alcohol, okay, hydroxyl. So we've used a hydrolysis here to split off number 4 over here from 1, 2, and 3. And we can keep hydrolyzing and taking off all those component parts. We can have exchange reactions. Exchange reactions, you're doing partially a synthesis and partially a decomposition just to swap partners. So here we have AB in this little blip, and then we have C over here, and we want to take this little blip off of AB and attach it to C. So first we have to hydrolyze this AB, right, to decompose it. And then we have to do a synthesis to add this little blip of B over to C. All right, so swapping partners involves decomposition first and then a synthesis. All right. All right, but the things mostly you want to care about is your synthesis and your hydrolysis. When we talk about chemical reactions and their reversibility, means that the reaction can go in either direction. So you start out with reactants and they're turned into products. And when the concentration of products gets real high, maybe above a certain threshold, they can convert back to the reactants, okay? And so we put these double arrows going in both reactions. We say the reaction has a chemical equilibrium, you know, that at a certain point, it's going to favor one direction or the other, depending on which part of the reaction is, you know, deficient. All right. All right. Each direction might need special conditions. Maybe you need to apply heat or something to go back to the reactions. You can write these things above the arrow over here. This will be a reaction that we'll see a lot with an enzyme. Carbonic anhydrase will act as a catalyst, and it will convert carbon dioxide and water into an intermediate carbonate product, and then you're going to have carbonic acid in the end, okay? All right, when we look at important biological molecules, we're going to talk about organic compounds and inorganic compounds. Organic compounds will always be built around carbon and hydrogen, often also with oxygen and nitrogen. Inorganic compounds typically lack carbon, or carbon is like insignificant. You can still see it in some inorganic compounds, and you almost get a feel for this after a while. But we'll be going through each of these macromolecules and figuring out, you know, what's a carbohydrate, what's a lipid, what's a protein. The inorganic molecule that we care about really is water. And like I said, water has all these special, almost magical qualities. This is why it's, you know, it's necessary for life. This is why when we send the Mars rover up on Mars, what are they always looking for? Is water, because if there's water, there can be life. All right. Water is inorganic, but all of our organic, you know, compounds need it, right? It's a polar molecule, as we've seen. It has this unequal distribution of charges, and it lines up in this unique way to have like a high boiling point, which allows it to be a temperature buffer. It is our universal solvent. So polar substances undergo dissociation in water. I have a picture coming up that shows this, what I mean. So all of our salts, and we're not really just water, we're salty water, right? When you go into the hospital, they don't just throw an IV of pure water on you, it's saline solution. The hydrogen bonds are able to absorb heat, okay? That helps with that temperature buffer. And it takes, like I said, all those little hydrogen bonds in between all the molecules. It takes a lot of heat to separate all the bulk because they're in mass, right? So this is what makes it be such a good temperature buffer for us. And you'll see that water is the reactant to our product in lots of the chemical reactions in our body. So that mass of all the numbers of those hydrogen bonds or hydrogen associations are what will allow water to absorb and release heat that allows us to dissipate it as sweat, right? And water is going to moderate then temperature changes. You know, all these chemical reactions giving off a lot of heat in our body, we could burn up without this. And as the energy inputs are going to continue, the balance between hydrogen bond formation and breaking swings back and forth, okay? And this ends up releasing the individual water molecules on the surface as evaporation, part of our sweating in that sense. Okay, so let's just look at this. Okay, so there's our polar water molecule. Now, when we have all these water molecules, why sodium chloride, a really strong bond, takes lots of joules of energy to, you know, burn up sodium chloride and break apart the bonds. But in water, they dissociate the particles, the cation and the anion from each other because water is polar. All right. So here's your lattice of the sodium chloride crystal we went through before. And in water, we have our sodium cation, and then all of our water molecules line up around it and keep it separated away from the chloride molecule, where all the polar end of all the positive protons or hydrogen delta pluses line up around that. So it keeps the sodium and the chloride apart from each other. And that's what is dissolving. Okay, that is sodium chloride put into solution. Because water is polar with all the slight positive and negative charges, it's going to surround all these sodium and chloride molecules. This is solvation. It's a sphere of hydration around each anion and each cation, okay? And it's that polarity of the water molecule that makes it this effective solvent for all living things being dispersed in water like that. All right, so let's try this. In a water molecule, oxygen forms hydrogen bonds with 2 hydrogen atoms to make an individual water molecule. Is this true or false? Is it tricky or not?
True.
True.
Water forms 2 hydrogen bonds with 2 hydrogen atoms. Okay, so let's look at this. All right, so if I were to take my hydrogen and my oxygen, how is hydrogen combined with oxygen?
A covalent bond?
A covalent bond. Is that a hydrogen bond?
No.
No. So here, in one individual water molecule, the oxygen is covalently bonded to the hydrogen, right? The hydrogen is covalently bonded to the oxygen. But if we have another water molecule over here, also where the hydrogens are covalently bonded to the oxygen, the hydrogen bond is between the hydrogen of one water molecule over here, right? And between a totally different water molecule over here. So is this question tricky?
Yes.
In a water molecule, oxygen forms covalent bonds, right? Covalent bonds.
Are there other types of molecules that form hydrogen bonds?
Yes, we'll go through those. We're just focusing, we're getting it down with water here. Covalent bonds with 2 hydrogen atoms to make an individual water molecule, okay? So that's false, all right? All right, but if I asked on that question, if a water molecule forms hydrogen bonds with another water molecule, then it would be true. Okay, so it's 11:30 and finishing on this with water is a good stopping spot. I got to jump in the car and drive out to Ann Arbor. So those of you who are going to see me today, we'll meet there today at class, and everyone else, we will pick up on Thursday, and I'll see some of you on Thursday. All A's this week, right? Anybody who's A group, and I graded all your little orientation things, and I told everybody if you're A group or B group, and hopefully I didn't make a mistake. Okay, and if you're sick, I had a couple of people write to me saying they're sick or they tested COVID positive. Don't come. We all don't want COVID, and you need to get well. I put the old online bone labs that I did in the past on online, and you can follow along with those. Okay, today we're going to start working on the bone labs in person. Those of you who are in the B group, you might want to look at those old labs and, you know, the slides to help you start filling in while you're sitting at home this week, some of those lab questions, so that you're better prepared when you come to look at the bones next week. Okay.
Real quick, where can I find those labs? What section are they going to be under?
The labs here, let's bring up Blackboard.
And can you remind me of the room that you're in?
254.
254. Okay.
All right. So can you guys see Blackboard, or I probably have to share it again, new share. All right. So you go into labs for Blackboard, labs for mixed mode. When you get into the labs, we're going to be doing bone labs. So lab 3, skeleton, axial skeleton. You come in here, and I have a list of bones, or Susan Dental wrote this list. All right. Lab 3 is your connect. And then there's all this folder here that says Grossman Manual Lab. That's what we're doing in person, or what you're working on at home. So you go in here and you click onto this PDF, and it will bring up the lab that we're working on today, lab week 3. All right. Now, underneath here, when we did lab at home, this is the last time I was doing lab at home, and I do the same lab whether it's at Wayne County or at Washtenaw County. So I think this is with my Washtenaw folks, as I recognize the people in the picture there. These are the slides that go along with this presentation, these pre-lab slides. Then I put in here some bone videos. So for when you're at home, you can go through this and look at videos of the, you know, skull and cranium. You can hit on this,
The
and
cranium is the part of the
it'll
skull
go
that
through
encloses the brain.
and point out the whole
It's made up of
bones. So those will be helpful for when you're at home. All right, and then here's more axial skeleton, atlas, all the vertebral column in that. And I have the same setup for lab 4. Okay, so if you go back in here, because we're working on lab 3 and 4, both at the same time over the next 2 weeks. So lab 4 is the appendicular skeleton, you know, your arms, your legs, your pelvis, and all that. So same thing, you go in here for the Grossman manual, you click on this PDF, and you'll get the PDF for lab week 4. You have this video here that goes over that PDF. So if you're sitting at home this week, you might want to watch that and start working on those answers. And here's, you know, pre-labs for that. I think I have 2 here. I have one that I did with people at Washtenaw and one that I did with people at Wayne County. So you can take your pick, whichever one you want to do. All right. Okay. Yep, Noah.
Thank you so much.
I know I saw your email about it too. So I am in uneven number, and I'm also in B.
Yes.
So
you
am I
come
in today, or
if group A is coming this week, group B is coming next week in person. Group B this week is working on maybe answering some of the questions on this lab, and, you know, maybe watching these videos along with everybody who's sick.
okay, gotcha. So I'll be
Okay. So if you're
on.
even numbered, that's group B come February 1st and February 3rd. So if you're a Tuesday lab and you're group B, you come next Tuesday. If you're a Tuesday lab and you're in group A, you have an odd number, you come today. Somebody will mix it up and it's okay.
Okay.
All right.
Okay. I'm trying. Okay.
Yeah. If you know, some people will mix it up and don't worry, we'll straighten it out in the end.
Okay.
All right.
All right.
Everybody good?
Thank you. Yeah, thank you so much. Have a good one.
You too. I'll see you Thursday and we'll finish those macromolecules.